Hi everyone, my name is Serena and welcome back to yet another video on my channel, Serena Kahani. Today I'll be doing something a little different and starting a new series which I'll be calling Biology in Resolution. So, as someone pursuing a biology related major in college, specifically biomedical engineering, I'm really passionate about topics centering around medicine, biology, chemistry, etc. However, I feel that when biology is taught in schools, it's often taught as a bunch of facts that are supposed to be memorized, but really, I don't think it's like that. Rather, biology is a bunch of ideas and concepts derived from decades and even centuries of experimentation and research, and all these concepts and ideas come together to tell a larger story. And I want to tell that story and hopefully form community and collective interest around it. And that's what the purpose of this new series, Bio and Resolution, will be all about. However, for today's video, I'll be focusing on proteins, which are kind of like the basic blocks for life and how we function, and really a lot of the stuff that goes on in our body. And this will be the first part of a two-part series on proteins, which I'll be calling Unraveling the Story of Proteins. And in this video, I'll be focusing on the first part of that story, how proteins are created. So without further ado, we'll get started. So first, I'll be going over how and why proteins are important. When we think of proteins, we probably think of what we eat: legumes, meat, eggs, etc. And these foods contain amino acids, which are what we use to build the proteins in our body. And the proteins in our body are used for many different things and take various forms, such as enzymes, which help digest food, and the tumor suppressor protein P53, which is essential for regulating DNA damage. However, how do we get to this point? Well, that's what we'll be answering in this video as we unravel the story of proteins, focusing on how they're produced. So it all starts with the genome. DNA, our genetic code, contains all the instructions in the form of nucleotide bases for making these proteins. However, DNA cannot leave the nucleus to be translated by ribosomes, which are little macromolecular machines found in our cells. So DNA must be transcribed into messenger RNA or mRNA, which is capable of leaving the cell.
As shown, a specific gene, which codes for a protein, will be transcribed into an mRNA strand. mRNA is single-stranded, while DNA is double-stranded.
However, not all of DNA codes for a protein. In fact, only 1% codes for proteins, and the other 99% codes for regulatory sequences. In addition, there were about 20,000 genes in our genome and about 100,000 distinct proteins. And this is due to the concept of alternative splicing, introns, and exons.
Introns are parts of DNA that don't code for a protein, while exons are sequences that do code for a protein. When mRNA is produced, introns are usually split out, as shown above. Yet, with alternative splicing, different sets of exons could be stuck together in order to create a variety of protein sequences. A couple of modifications are made to mRNA to extend its life in the cytoplasm. A methyl cap is added, and a long sequence of A nucleotide bases, known as the poly-A tail, are added to the end of the mRNA sequence. The cap assists with mRNA binding to ribosomes, and the poly-A tail preserves the actual content sequence, as in the cytoplasm, nucleobases, or enzymes that degrade nucleotides, will degrade the poly-A tail instead of the actual sequence directly. Now, once in the ribosome, the mRNA will be translated into a protein. mRNA is read in groups of 3 bases, called a codon. In essence, mRNA enters the ribosome through one end, and transfer RNA, or tRNA, brings the corresponding amino acid to build a protein. Eventually, the mRNA will exit the ribosome, leaving the finished peptide chain.
However, what's interesting is how the protein folds, as protein structure determines its function.
A peptide chain, made up of several amino acids, forms the primary structure. Hydrogen bonding between the peptide backbone causes the chain to fold in a repeating pattern, forming either an alpha helix or beta pleated sheet known as secondary structure. After, a 3D structure takes shape due to side chain interactions. Specifically, ionic bonds and disulfide bridges form between the R groups of the protein. All proteins on Earth have tertiary structure. For a protein to have quaternary structure, protein subunits must come together. Because protein structure determines function, elucidating the structure of proteins is a key part of biomedical research. For example, the digestive enzyme amylase is a chain structure that allows for optimal binding to starchy substrates. Meanwhile, hemoglobin contains inorganic heme groups which bind to iron, allowing it to transfer oxygen to the rest of the body. Yet, there are many proteins for which the structure is unknown, part of what we call the dark proteome. To uncover the structure of proteins, scientists will use techniques such as X-ray crystallography, which is quite expensive. Now, techniques using artificial intelligence and deep learning have been employed to predict the structure of unknown proteins. One of these innovations include AlphaFold 3, developed by DeepMind, a subset of Google. This has been highly effective at predicting the structure of proteins it hasn't seen before.
The process to produce a protein from start to finish is incredibly complex, and honestly, it's pretty amazing that our bodies can do all this. However, mistakes can happen. There can be mutations in the genetic code leading to the production of a faulty protein, or a protein may fold incorrectly, leading to some pretty dire consequences. This is what we'll be covering in the next video of the series and the part 2 to this video. We'll be going over the results of protein production and the impacts that they have. So with that being said, thank you so much for tuning into this video. If you enjoyed this video, I encourage you to give the likes up and to please subscribe to my channel. And if you would like to see more videos in the series, Biology and Resolution, please do let me know, as honestly, they're pretty fun to make.